Dr. Karen Parker is the Trong Tan Broadcom Professor and Associate Chair of the Department of Psychiatry and Behavioral Sciences at Stanford University, where she leads the Major Laboratory Steering Committee and directs the Social Neurosciences Research Program. The principal goal of her work is to better understand the biology of social functioning across a range of species and to translate these fundamental insights to drive diagnostic and treatment advances for patients with social challenges with a core focus on autism. These webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website at autism.org. And now I will turn this over to our speaker.
Thanks, Denise. It's a pleasure to be here with you all today. Today I'll be talking to you about my lab's translational autism research roadmap. And so this is sort of an overview of the work that we'll be discussing. So I'll talk about the need for and development of a valid animal model to identify biomarkers and targets, our work to translate this work from our animal research to children with autism, and then a clinical trial that we've conducted in children with autism, as well as ongoing research. So I think that everyone here is familiar with autism. It's characterized by two core features, persistent social interaction impairments and the persistence of restricted repetitive behaviors. Unlike other medical disorders that have a simple scan or blood test, because we don't have a biomarker, which is an indication of a disease state, we have no laboratory-based test, and so diagnosis is behavioral. Currently, there are only two FDA-approved medications, which are both antipsychotics that treat associated features like irritability. They have negative side effects, and there are currently no medications for the core features of autism. I want to point out very early on that this lack of medication options for autism stems from a drug development crisis for brain disorders more generally. So there was a semi-recent paper in JAMA indicating that it takes about $1.3 billion to bring a new drug to market. About 90% of central nervous system or brain medications fail, having been efficacious in preclinical models. And about 50% of those medication failures can be attributed to poor animal model selection. And so we saw that there was an opportunity to develop a refined animal model with high translational utility. I want to point out that not all models are wrong, but some are useful. And so every single animal model can be useful in its own way, but we have to think really carefully about the criteria that will be best suited for what our goals. And so when I started thinking about animal model development, we need to think about onset. So if we're modeling autism, we want it to be childhood onset. If we're thinking about dementia, we want to have later adult onset. We also want to think about prevalence. So in autism, about 3 to 4 males are affected for every 1 female. If you're modeling depression, it's 1 male to every 2 females. We also want to think about characteristics that are either homologous or endophenotypic, which essentially means, are we studying the actual disease or disorder in the animal, or are we studying a piece of it? And so something like diabetes, you could have an animal model where the animal has diabetes. There is not good evidence to suggest that you can study autism in an animal model. And so the work that we started thinking about was modeling specific features. We also want to be able to establish face validity, which is that there's an outward similarity in appearance between the model's attributes and the patient's symptoms. So in the case of autism, we would want complex social cognition difficulties and repetitive behaviors in a highly social, diurnal species with vision as its primary sensory modality. We would also want to establish construct validity, which is that we can observe similarity to the underlying causes of the disease or disorder in the animal model and in the patient population of interest. And work to date had focused on, say, large neuroanatomical brain lesions But what we wanted to do was think about measuring homologous genes and circuits. So things that would be naturally there versus environmentally induced that really don't represent the causes of autism. We also want to establish predictive validity, which is the model's ability to identify and evaluate drugs for safety and efficacy. And I just want to note that the cautionary tale of thalidomide, which was a morning sickness drug that was tested not in primates, and it was shown it was not tested in primates. It induced gross limb abnormalities in people with in babies that after they had been born. And so when researchers went back and looked at toxicology in primates, we could see these limb abnormalities. So what we really want to do is make sure that the model allows us to have reliable readouts in the animal model and in the patients. And so then when we think about animal model development, we want to think about model features, and you want to weigh the pros and cons. And so there are certain things that would be easier to do in rodents versus in primates, but there might be higher payoff to doing primate research. And so we were interested in thinking about primates because they are more closely related to humans, and so they would have more evolutionarily conserved gene expression and neuroanatomy. And also for the reasons I mentioned with thalidomide, potentially more streamlined safety and efficacy endpoints. So when we started thinking about the points of entry and validation for our model, as I mentioned before, there's these two core features of autism. I also want to point out it's highly clinically heterogeneous. And so we could have a genes first approach where we might identify homologous pathogenic variants, so specific genetic variants that are causative for autism. We could do selective breeding or genome editing, which was really expensive when we first started this work. We could have a behavior first approach that would allow us to establish face validity. So looking at naturally occurring behavioral features like social interaction difficulties and repetitive behaviors. And/or we could think about looking for biomarkers. So we could perform biomarker discovery in the animal model and then translate to people with autism. And so we started our work thinking about a behavior first approach and then began validating with biomarker discovery. So when we look to the human genetics work, although there's been a lot made of single gene mutations in autism that are highly rare but highly penetrant, the vast majority of genetic burden in autism is polygenic inherited. And this observation of polygenic inherited risk can, I think, be neatly illustrated by this graph from work that John Constantino did in the early 2000s, showing the distribution of quantitative social and autistic traits in the general human population. And the idea is that this polygenic inherited autistic trait burden underlies this quantitative variation in social traits. And so what we wanted to do was to develop methods to identify and study individuals at the quantitative extreme of social functioning in a general monkey population. So this was our strategy. So this is the social responsiveness scale, like I mentioned, and I'm not going to bury the lead. This is the approach that we ended up using for a lot of our work. So this is quantitative social and autistic traits in the human population, with autism representing this right tail here. And then in our animal model, we were able to reverse translate this scale and then be able to demonstrate we had a very similar distribution, and then begin studying the monkeys that have these naturally occurring social impairments that are at the extreme of this population. And then what we could do is in our animal model, begin to perform biomarker discovery and then translate this to people with autism. And so we were interested in studying animals that were as closely related to people as possible. And a very common lab animal model are rhesus monkeys that diverged from humans about 25 million years ago. And there were other attractive features. They're highly social like humans. They have complex social cognition abilities. They do use vision as their primary sensory modality. They show stable individual differences in social behavior. There was evidence from the literature that there were animals that exhibit naturally occurring social impairments. And the nice thing about rhesus monkeys is that roughly that every 1 year of a rhesus monkey corresponds to about 4 years of human life. And so we could begin to study development in early adulthood in a fairly compressed period of time. I like to joke that Stanford wouldn't let me put, 4,000 monkeys on campus. So I had to take my show on the road, and we went to the California National Primate Research Center, which is about 100 miles from my campus. And the rationale for this was that these monkeys were housed outdoors in half-acre corrals. And so if we were seeing spontaneous social impairments, they were likely to be naturally occurring versus environmentally induced. This large cohort would mean there would be more monkeys at the extreme to be able to study. They are housed outdoors in naturally occurring situations that resemble their natural ecology of mixed male-female social groups of about 90 to 225 animals. We could study all ages and study across the lifespan, and genetic pedigrees were known if we wanted to be able to move our work into the genomic space. And so I had two really terrific postdocs in the lab who helped develop 3 different ways of identifying these that monkeys that exhibit naturally occurring social deficits. One is a brute force focal animal sampling, where people stand outside of the cages. This is people on our team, and the monkeys are very used to people being outside the cages. And so using a rhesus macaque ethogram, we could quantify naturally occurring social behavior. As I mentioned before, we could reverse translate this social responsiveness scale that has the dual function in people of not only being able to measure quantitative variation in social and autistic traits, but it can actually be used for a screener for clinical referral for an autism diagnosis. And then we developed a variety of different tests that we could use to study features that clinicians had told us were important in terms of thinking about autism. And what I want to say is that the rest of the work that I'll be presenting is looking at monkeys at the extremes of this distribution using this focal animal sampling method. And so for the past now, over 10 years, we've been evaluating the behavioral features of these monkeys that are relevant to human autism to establish face validity. And so what we found was that these naturally low social male monkeys show this greater burden of autistic-like traits. When we test them on laboratory tests, we bring them inside and administer tests. They show abnormalities in species typical perception and reaction to social stimuli. So they're able to recognize objects, but they do fairly poorly on face recognition. And they do poorly on species typical responses to videos showing social cues. So they don't react like typical rhesus monkeys to, say, an affiliative overture. They don't react and reciprocate that sort of behavior. When we did observations outdoors in the home corrals, we saw impairments in reciprocal social interactions. So in this species, it was diminished affiliation and grooming. And we saw decreased initiation, but not receipt of pro-social behavior, suggesting a social motivational deficit. I had a really great vet student in my lab one summer, and he did a medical record review. And what he was able to show was that low social monkeys have a greater number of traumatic injuries and bullying by their peers. And much like what's seen in epidemiological studies in the autism population, this worsens with autistic-like trait burden. And then we were also able to show looking at there was at the time this wonderful screening program to screen all infants at the California National Primate Center. And we were able to pull out tests in that battery that allowed us with 100% accuracy to predict which infants would go on to be low social later in adulthood, suggesting that we could identify a period of time where these animals have subtle social information processing deficits where we might be able to intervene later. Um, so then the next thing we wanted to ask was how could we biologically detect social deficits? So I was interested in examining fluids which would enable direct translation from our monkey model to people with autism. And I want to point out that autism is largely viewed as a brain disorder. And the vast majority of work that had been done in the past was using blood. And blood is easier to get, but the studies had poor detection and poor reproducibility. And a fluid called cerebral spinal fluid, which bathes the brain and spinal column, is more representative of brain biochemistry than blood, but it does require a lumbar puncture, which is a more invasive procedure than a blood collection. What I had found early in our research and that I found quite interesting was that neurologists had been using CSF to perform biomarker discovery in their neurodegenerative disease, and that it could be useful for identifying the first biomarkers. And this was the case for both Alzheimer's and multiple sclerosis. So we wanted to see if we could use a similar approach in approaching autism. And so what we did in this first study was we selected all available male monkeys that were ages 2 to 5. We performed hundreds of unobtrusive quantitative assessments in the monkeys' home field corrals to identify these low social monkeys. And then we quantified candidate CSF and blood-based biomarkers. And what I want to point out here is that the biomarkers we selected in this initial proof of concept study were any pathways that had been associated with mammalian social behavior, any with idiopathic autism, and then any that were associated with genetic syndromes. And so I was particularly interested in arginine vasopressin, or we'll just call it vasopressin, and oxytocin, which are these ancient molecules. They're both 9 amino acid peptides, and they only differ by 2 amino acids. And they had been implicated over and over in every mammalian species studied to be involved in social functioning. And then these other kinase signaling pathways had been implicated in rasopathies, which had high penetrance for autism. And so we were also interested in studying these. So then what we did is we had our low and high social monkeys at the extremes in this initial first cohort, and we could put all these measurements in a discriminant analysis and simply say, can you tell us who is high and low social? And the answer was yes, with fairly high accuracy at about a 93% correct classification. But this didn't enable us to determine what molecules were important. And so then we performed a logistic regression, and we were able to identify CSF vasopressin, as well as 2 endpoints in these kinase signaling pathways that additively allowed us to classify these individuals. But if we're thinking about a biomarker, we would also want it to differ significantly between groups. And the only biomarker in this biomarker winnowing strategy that we implemented was cerebral spinal fluid levels of vasopressin. So then what we did is in multiple follow-up cohorts, we collected cerebral spinal fluid vasopressin. And I'm going to walk you through this graph because we use it repeatedly, both in our monkey work and in our work with people with autism. So this dotted line represents a 50% correct classification. And these blue is always low social or autism in all of my slides. And red is either are basically the controls of that given species. And what you can see here is that monkeys that were above the dashed line were correctly classified in blue, and any below the dashed line in orange were correctly classified. And so by just knowing CSF vasopressin concentration, we could, with very high accuracy, classify animals as high or low social. We were also able to show this robust group difference. We looked to see if spinal fluid levels of vasopressin were related to a really critical behavior that rhesus monkeys and many other primates perform, which is social grooming, which allows an animal to both initiate social bonds and maintain them. And we had a very nice correlation such that the animals with the lowest CSF vasopressin levels uh, showed the least amount of time in social grooming. We also looked in just the general monkey population at whether vasopressin levels in spinal fluid were related to autistic trait burden using the macaque social responsiveness scale that I mentioned previously. And this is a reverse scored instrument. So here again, we show that the animals that had the lowest CSF vasopressin concentrations had the greatest autistic trait burden. And then, if you're trying to demonstrate that something is a biomarker, you need to be able to say, well, if I measure it at time one, do I see that the animal has about the same concentration of vasopressin if I measure it across time at different time points? And so, here, what you can see, we removed CSF on 4 different occasions. Each of these ticks on the x-axis is an individual monkey. What you can see is there is a very large variation in spinal fluid levels of vasopressin, but within a given monkey, we see a very restricted range, suggesting that there's trait-like consistency of vasopressin in these animals. What I want to point out is that there's been a lot of work done on vasopressin and oxytocin, and oxytocin has received probably the majority of attention. But what's interesting is if you go back to the literature, vasopressin was shown to be critical in regulating male social functioning, which I found interesting because this is suggestive of a sex vulnerability potentially, and is consistent with this male bias vulnerability for autism. We've been able to replicate this multiple times in independent monkey cohorts, so it's a reproducible finding. These group differences in vasopressin were not ever evident in blood. So if we'd only looked in blood, we wouldn't have found this. So it suggests that it's brain specific. And in none of our studies have we ever seen group differences in either CSF or blood oxytocin levels, which is pretty striking because these molecules perform similar functions. And like I mentioned previously, they only differ by 2 amino acids. And so oxytocin has actually become a negative control in our ongoing work. So a lot of the feedback that I got when we started presenting our monkey work to clinicians was, well, low social monkeys, that's a non-pathological entity. And so what does this ever say about people with autism? And so we were interested in directly taking our findings from our animal model to applying them in a clinical setting. And so we were very fortunate to partner with multiple pediatric clinicians who were able to identify children undergoing spinal taps for clinical reasons. And then we were able to get additional CSF with research consent to be able to have to look at children that had autism and children that were controls or socially intact. So they didn't have autism. And then all these kids were rule out negative for the reasons they were having an LP performed in the first place. And so in our first study, what we found was, again, this is very similar to what we did in our monkey work, the 50% dashed line. And any people with, these are children with autism that are correctly classified, and those that are correctly classified that were in our medical control group. So we only had one person misclassified on the basis of their CSF vasopressin concentration. And again, like in our monkey model, we were able to see this very robust differences between groups. Subsequent work that we did, we were really fortunate to be able to team up with Sue Sweeto, who's was at the time an NIH researcher. She's since retired. And she was doing some work with research indicated lumbar puncture. So these were not kids that were undergoing a lumbar puncture for clinical purposes. They were being enrolled in her research studies. And so these were very well characterized children. And so we were able, and this is the largest CSF cohort to date, so we were able to ask the same question in children that were medically healthy. And we were able to show that there were, again, this group difference in CSF vasopressin levels. This was the first time we were able to include girls in our studies. And what was interesting is I had sort of thought, well, maybe we'll see an oxytocin difference in these girls. And the answer was no. Both in the males and females with autism, we saw diminished, statistically significant decreases in vasopressin, but not oxytocin. And we also saw a very nice correlation between CSF vasopressin levels and symptom severity on the ADOS, such that when we decompose that clinical symptom severity, the vasopressin was tracking the social symptom severity, but not the restricted repetitive behaviors, suggesting that there might be other biomarkers that are tracking that piece of the phenotype. And then I mentioned that we didn't see any differences in CSF oxytocin. So then we did a follow-up study in just a simple blood cohort with a large number of children with autism and matched neurotypical controls. And again, just like in our monkey model, we saw no group differences in blood. And so this again indicated in people with autism that this finding was specific to CSF vasopressin. And so this is some unpublished work from our group. We have recently been looking at postmortem human brain tissue, and vasopressin is made in the hypothalamus. And so what we wanted to ask was, obviously, if we're thinking about the clinical significance of this, we would need to show that the CSF vasopressin was a readout of where it's made. And so what I want to point out here is we've done work in both ante versus postmortem population, in kids versus adults, using the ADOS and the ADIR, which are both research diagnostic assessments for autism. And we've even used two different ways to detect vasopressin using an ELISA versus mass spec. So in this follow-on study from postmortem tissue, we again were able to replicate this nice relationship, showing again that the lower the vasopressin levels, the greater the social symptom severity. And then we were also able to show that CSF vasopressin levels were robustly correlated with postmortem hypothalamic vasopressin gene expression, but this was not the case with blood, really driving home the point that the CSF is the signal where we see vasopressin. Okay, so the next study that we did was we were very interested in thinking about how early could we detect this vasopressin deficiency. And so is it evident before symptoms first manifest? And so I teamed up with John Constantino, I knew that he had an infant CSF cohort of well over 2,000 samples. And so he agreed to collaborate. And so we were able, he had taken these samples and stored them at minus 80 in a freezer. And then what we had to do, this was before there were electronic medical records. So John's team heroically traced the paper medical records to the electronic medical system. At the time he was at WashU. And then what we were able to do is, in a quasi-prospective way, follow these babies. So they were newborn human infants, and then we followed them in the medical records through 12 years of age, and were able to identify a subset received an autism diagnosis and a subset that diagnosed that proceeded to develop typically. And what we were able to see was that in these newborn humans, we already had statistically lower levels of vasopressin in those that went on to receive an autism diagnosis many months to years later. And this was specific to vasopressin because, again, we saw no difference in CSF oxytocin. And to me, this suggests that if we're able to identify infants, particularly maybe those at familial risk, if we're able to identify individuals that we think are at high risk of developing autism, we might be able to intervene behaviorally before symptoms manifest and more radically alter developmental trajectories. So the next thing that we did was we were interested in thinking about treatment. And so we knew that vasopressin was a robust biomarker of primate social impairment. We were able to, again, show this in people. And there had been one of the first receptor mapping studies done in rhesus monkeys showing so vasopressin's receptor in the brain that's been associated with social functioning is the V1A receptor. And this unlike the oxytocin receptor, is densely distributed throughout the primate social brain, suggesting that if we administered vasopressin to people with autism, that we could directly target social circuits. And so we asked this question, does vasopressin treatment enhance social abilities in children with autism? And so we know that if you give intranasal vasopressin, that it penetrates the spinal fluid. And that was a study done a long time ago by a European group. And that we don't exactly know how intranasal vasopressin works, but what we do think that there's several ways that it could. It could enter the brain through nasal epithelial or potentially traveling along cranial nerves like the olfactory nerve or the trigeminal nerve. We do know from brain imaging studies that there are robust changes in bold signal on fMRI after neuropeptide administration. We also knew at the time that single doses of vasopressin in healthy male adults enhance memory for social information, identification of social words, and increased cooperative behavior, suggesting even after a single dose, we could see prosocial behavior in neurotypical adult individuals. But no prior research had tested the safety or efficacy of intranasal vasopressin to improve social abilities in children with autism. And so I teamed up with Antonio Hardin, who is a child psychiatrist and a clinical trialist with specialization in autism. And we conducted a blinded, randomized, placebo-controlled trial of vasopressin that was 4 weeks in duration. And so we tested safety and tolerability by examining steady dropout rate, vital signs, clinical chemistry labs, electrocardiogram, and patient-reported adverse events on an instrument called the DOTS. And then what we also did is after 4 weeks of treatment, we used we assessed social abilities, and we did so using the social responsiveness scale, which is what the FDA asked us to use. But as a basic scientist, I like to see things and I like to see convergent validity. And so we also evaluated improvement in social behavior by clinician evaluation on the CGI, and then also child performance on laboratory-based social cognition test using the reading the mind and the eyes and an affect or motion recognition test that we developed. And so these were our methods and procedures. We had this was a pilot trial, so we had 30 children with autism, age 6 to 12, with IQs above 50, social responsiveness scale scores above 70, CGIs above 4. We verified a historical and DSM diagnosis with both the ADOS and the ADIR, which, as I mentioned before, are research diagnostic assessments. And then we used two different doses in a younger group and an older group. And the dose actually was not statistically significant, but this is the dose we used. And at the time, the FDA had asked us to use an IV injectable Vasostrict, which is approved for cardiac shock. And so we transferred this to nasal delivery vials and then compounded an identical placebo. And then we assessed drug safety and tolerability weekly. And then we evaluated efficacy before and after the 4-week treatment trial. So we saw no vasopressin treated patient dropped out of the study. We saw no differences in adverse events between groups. And we saw no significant changes from baseline in patients' vital signs, clinical chemistry labs, or electrocardiogram during vasopressin treatment. And then in terms of efficacy, we saw that parents, so these are all, blinded parents, blinded clinicians, blinded children. We saw improvement in social responsiveness scale scores based on parent ratings. We saw a statistically significant increase in clinician impression. And then we also saw on child-based lab test that children perform better on the reading the mind and the eyes test, as well as on affect recognition following 4 weeks of vasopressin treatment. So around the time that we were doing this work, Roche had come out with a compound called balovaptin. And unbeknownst to us, when we went to publish this, our study in Science Translational Medicine, the editor also rolled out the balovaptin study in the same issue. And part of the reason why this was really interesting was we were using diametrically opposed approaches. So we were using a vasopressin agonist, and they were using a vasopressin V1A receptor antagonist. And so our rationale was that vasopressin, as studied in social rodent species, induces pair bonds or attachment relationships and promotes paternal care. And we know that if you give a vasopressin V1A receptor antagonist to these social species, it impairs social behavior in the presence of normal oxytocin signaling. So it's not like oxytocin doesn't take over the role. So it's very specific to vasopressin, this pro-social role. There was also a rat called the Brattleboro rat, which had a mutation in its vasopressin gene. And these rats showed a social impairments as well as restricted repetitive behaviors. And then, of course, we've already reviewed all of the low CSF vasopressin work in both our monkey model and in people with autism. And also both in our animal model and in people with autism, we saw that the lower the vasopressin levels, the greater the social impairments or symptom severity. And so we were never able to get a really good read on why Roche had taken this take. One possibility, because they were a company, they didn't have to report their whole line of reasoning the way that we did. And so we thought that it was possible that maybe giving a vasopressin V1A receptor antagonist might make oxytocin receptors more accessible. There was some evidence in the literature that giving vasopressin induced flank marking, which is a sign of territoriality and aggression, and what I'll call neurotypical asocial gold enhancers with intact vasopressin signaling. And this was a species that doesn't engage in social behavior. The only things in its repertoire are mating and territory guarding. And so, when you give vasopressin to one of these animals, it doesn't have the sort of range of behaviors that, say, a very social prairie vole would. And I think this comes back to the point I made at the very beginning of our study, at the very beginning of my talk, which is animal model selection is incredibly critical to provide insights into patient work or not. And so the balovaptin work has since been terminated due to an interim futility analysis. So my understanding is that compound has been shelved. But at the time, we were really interested in saying, let's look at some of this stuff in our monkey model. And so what we did is we had monkeys, we developed a voluntary nebulization protocol where monkeys would drink a favorite juice or yogurt, and they would put their nose into this nebulization cone, and then we could precisely administer on 3 different time points either a placebo, a low or a high dose of vasopressin. And the points I want to make here are we studied the behavioral effects. And so what we did is we administered a recognition object memory test as well as a face recognition test. And then we showed these monkeys videos of a conspecific monkey either engaged in affiliative behavior or aggressive behavior. And then we recorded the subject monkey's responses. And then we also did a pharmacokinetic study because this was a novel way to administer vasopressin. And I'll just give you the take-home message, which is that we saw that vasopressin penetrated spinal fluid in our study, given through nebulization, suggesting that it penetrates the central nervous system. And so then what we did was we gave single doses of vasopressin, and what we had was a visual paired comparison recognition memory test. And if you remember, I had mentioned that these low social monkeys have intact object recognition, but they don't do very well on face recognition, which is something that has been observed in at least some studies of people with autism. And so what we did was we administered, so this is comparing, there were no drug effects. So we've collapsed the high and low drug doses here. And so if you look at the placebo-treated monkeys, that they show no face recognition memory at baseline, but they do object recognition memory. And what's interesting is that what we see is after vasopressin administration, the face recognition ability is rescued, and it is indistinguishable from their object recognition memory, either on vasopressin or placebo. And then the next thing we asked was, well, and I think I had mentioned to you that when we do these outdoor observations of these monkeys, we've shown these sort of social motivational impairments. And so we basically asked, if we show you a video of a monkey behaving affiliatively, rhesus monkeys will lip smack, and there's a whole repertoire of affiliative behaviors they will do. And these low social monkeys don't really respond. But what was interesting is here what we see. So this is on placebo. We saw no response to affiliative cues. But once we treated them with vasopressin, the low social monkeys began responding to these affiliative cues, suggesting that they could reciprocate these social overtures. And then, I was always very skeptical of this aggressive behavior, especially because it had been tested in individuals that had intact vasopressin, and they had never been tested in arguably the right species. And so so what we did was we gave our monkeys either placebo or vasopressin, and either on placebo or on drug, they did not respond aggressively to aggressive overtures, suggesting that vasopressin was selective in altering prosocial but not agonistic behavior. Okay, so there's a lot of unanswered questions. The first question I had was, could we use a vasopressin V1A receptor to block these improvements in face recognition and reciprocal affiliative responses? I asked Roche repeatedly for their drug, but they wouldn't give it to me, so we weren't able to run that study. And then one question would be, if we gave vasopressin to what we'll call the neurotypical high social monkeys with intact vasopressin, would this have made them aggressive, or is this really only a finding in rodents and it doesn't apply to human and non-human primates? In our first clinical trial, we saw no evidence that vasopressin increased or moved aggression at all in children with autism. Other questions that I'd be interested in answering are, does vasopressin impact other dimensions of social behavior, really probing motivation? What are the effects of chronic vasopressin administration? Recall that these are only single doses, and we're already seeing these major changes. One study that I've always wanted to do was to give vasopressin to these baby monkeys with these subtle social information processing difficulties and see if we could administer it very early in life and radically change their developmental trajectories. So what I want to do is conclude on this work and then mention some of the work that we're currently doing. So hopefully I've convinced you that valid animal models are indispensable for understanding conserved biological substrates of social functioning, including the extremes of the social continuum, which includes autism in people. That these kinds of models hold promise for testing new medications with validated endpoints for streamlined translations. We could use the same endpoint in a monkey that we could in people with autism. I always like to point out limitations of our work. We have not yet studied repetitive behaviors, so we have only studied one piece of this phenotype. And we did do one study on female monkeys, but they look really different than males. And female rhesus monkeys are very hierarchical. And so when we look at dominance rank in males, it's orthogonal to these social impairments. In female monkeys, because dominance is so critical in females in this species, the dominance rank just swamps everything else. So I think this suggests we would either have to look for other ways to study social functioning in female rhesus monkeys, or female models of autism might be better developed in less hierarchical monkey species like, say, marmosets or TD monkeys. So we've done work showing that CSF but not blood vasopressin level is a robust neurochemical marker of human and non-human primate social functioning. CSF but not blood could be useful for detecting autism and risk for developing it. A couple limitations I want to point out are controlled children in at least 2 of the 3 studies were sick enough to require a lumbar puncture to begin with. Although our monkey work shows, our medically healthy monkeys, we show the same thing. And then the work we did with Sue Sweeto, at least, our children with autism were medically healthy. And the other thing is that we did not have a psychiatric or neurological control group, given how incredibly difficult these studies were to execute. And so what we don't know is vasopressin a marker of an atypically developing brain, or is it specific to autism? We know that vasopressin is well tolerated and improves social abilities in children with autism. I think it holds promise to be the first medication to treat core features. I'm often asked by behavioral pediatricians, I give desmopressin all the time for nocturnal enuresis. I don't ever see a change in social behavior. And what I want to point out is desmopressin targets the AVP, the vasopressin receptor 2, and it does not cross the blood-brain barrier. So limitations were that this was a very small sample size and requires replication, and we don't yet know the mechanism of action. I want to put in a plug for animal model research, which has been under attack in our country. What you can see here is this is a review article I published a couple years ago. And what we have taken upon ourselves, and I think one problem with a lot of the work that's done in this field is people will do animal model work, but they don't make themselves translate the work. And so they're under sort of no obligation to think about if the animal model they're developing will ever have translational utility. And I think that's how we end up having the problems that we've had. But what we've done is we've sort of taken it upon ourselves to directly translate anything we've done in monkeys or work we've done in humans and then reverse translate. So we've set up this forward and reverse translation that I personally think that all disease work should have. And then what you can see is there's a couple things we still don't know, but I think this is what can happen when you have a validated animal model for whatever you're interested in studying. Okay, and then I'm just going to end by talking about some of our ongoing research. We have been doing, I think I mentioned earlier in the talk that vasopressin really tracks social symptom severity. And so we've been doing some what I'll call CSF first biomarker discovery using a proteomics platform to identify proteins in spinal fluid to see if we can identify the signature there and then back it out into blood. And so that work is well underway, and I'm hoping we'll be able to submit it for publication this year. We're continuing to collaborate with John Constantino, who's now at Emory, to be able to do a prospective infant CSF collection consortium where we can ask, does vasopressin in another follow-up cohort predict autistic trait burden or autism diagnoses later in life? This one, so we have finished a follow-up phase 2B vasopressin clinical trial enrolling 108 children with autism. So this is the largest single site medication trial for autism to date. And that will read out hopefully this coming year. I'd be happy to come back to speak with you about findings of that in the future. And then I'll finish with something, this is sort of a message to the clinicians in the audience. After we started publishing our work and I appeared on a popular podcast, I was inundated by clinicians and parents of kids with autism, as well as adults on the spectrum, that there was excessive thirst and fluid consumption and urination. And this got us really to thinking about, could we think about the peripheral these peripheral symptoms that would be indicative of vasopressin insufficiency as a way to think about personalized treatments for people with autism. And so I wrote a perspective piece last year, and it's a pretty short piece. It's on my website, but we're very interested in doing an epidemiological study to look at these symptoms of vasopressin insufficiency and their relationship to autistic social impairment. And then what I want to do is just end by acknowledging all of the collaborators, mentees, RAs and techs, the families that have participated in our work, and all the funding that's made the work possible, and then you for your attention. And I'll stop here and take questions.
All right. Well, thank you so much for that great presentation. It was really interesting and so many questions. So I'm going to dive right in. We've got about 6 minutes, so I'll try to get through as many as possible and look for some common themes. People did ask about differences between males and females with vasopressin and what kind of research has been done on that, if any. So do you have any insight about that?
Yeah. So, I mean, the answer is that we had that one cohort where we studied the girls with CSF. That was the paper with Sue Sweeto's group. They look like the boys, at least from the CSF vasopressin perspective. Interestingly enough, the correlation between symptom severity in the males was far stronger, and in the females actually didn't even achieve statistical significance. In terms of the treatment trials, the last trial we just weren't sufficiently powered to look at sex differences in an N of 30. So I think I think it's really important that we do work with females and we try to recruit them as much as possible into our studies, but it can be difficult.
Okay. When you were doing this, I know you touched on this a little bit, but in a general way, how long did the effects of a single dose last when you were administering these in the trials?
So the single dose was only in monkeys, and we didn't evaluate that, so we just did the testing. And then we actually don't know. There were families who reported anecdotally that they thought that the treatment for 4 weeks had lasting changes. But again, that's not something we've studied. The current trial investigates up to 8 weeks of treatment. And then my hope is that we could do a phase 3 trial, a multi-site phase 3 trial where we can really look at long-term treatment.
So that leads into the next question. People are asking, how would I get in a trial? Like when that time comes. So is there somewhere they can sign up for emails or information?
Yeah, there's so my collaborator Antonio Hardin has a Stanford Research Registry, and that people could sign up for that. So I think when we have to find funding for the phase 3 trial, which as everybody knows is incredibly difficult under the current circumstances, and so we'll have to go raise money and then we'll have to roll out the trial. So we're currently looking into ways to fund this work. The current trial is closed, and so we aren't enrolling anybody anymore. We're just trying to finish the trial and get it published. So, stay tuned. If science prevails, we'll be able to get the money needed to hopefully push forward on this medication.
Okay, so this is a comment from a clinician. It says, it looks like the studies have typically lasted up to 8 weeks. Despite good safety profile data, recommendations have persistently included blood monitoring at baseline 4 and 8 weeks to watch for hyponatremia.
Yeah.
If this is requested by parents as a treatment, is this something they need to discuss with their doctor, or is this something they need to be aware of?
Yeah, I mean, I think that's something, I mean, it's a really good point. So we have not seen it to be a problem in either of our trials, but we've been very careful in screening people up front who had no sodium issues and had clear EKGs. So, if there are people with abnormalities, particularly if they have polydipsia, for example, I would argue those people were not included in our trials to date. So I don't really have any information about that. My hope is we'll be able to sort this out in a much larger trial.
So this may be off the wall. You don't have to answer this if you don't want to, but somebody did ask, it's an interesting question. How far would we be from AI models to work on this type of work? Have you heard anyone talking about that or seen anything like that?
I guess I'm not clear on if the AI models are animal models, or if the AI would be using like machine learning to identify biomarkers in,
Right.
substances. Yeah.
Yeah, they didn't clarify, so we'll see if they write back and ask some more. I think they were talking about looking at like social interaction, like facial interaction and that sort of thing. But yeah, that might be pretty complicated.
Well, there's definitely people that are doing that kind of work, right? Like where they're doing very dense sampling, because of the technology that's available now. We haven't done that, but I certainly have been at meetings where researchers have been talking about doing that.
Okay, I think we have time for one more question.
Sure.
And you may have answered this, so I apologize if I missed it. But do you see a correlation between the initial SRS score to the improvement in the score? So, in other words, was there evidence if the largest improvement in that SRS score was seen if someone initially scored a lower or higher SRS score?
Yeah, so my recollection is that, if you've got more to move, then we saw a greater impact. So I don't quote me. I think this is probably in our paper, but I want to say that people that were more severe, that had higher SRS scores moved more. But again, that was in 30 people. So, we'll have to make sure we look at that in the second trial.